Let's go ahead and get started. You can swap with me for 1 second. Hello, everybody. Thank you so much for coming, people in the audience and online. This is a great progress over government shutdown days.
Yeah,
Last
we might
quarter.
have another one.
George Cooper is our speaker. He was actually supposed to speak last quarter, but then the government shut down. So all of I think every single speaker we had lined up were from NASA Ames because of the Space Act agreement. So it just put a damper on those plans. But here we are. We're back. No more government shutdown. We're really happy to have Dr. Cooper here with us. I want to give a few announcements before we start. First, I want to give kudos to David Deemer, who is here. Last quarter, I think it was last quarter, you were honored with the Drake Prize. I believe that was in October.
Yes, that's right.
Yes. So the SETI Institute issues the Drake Prize. I don't know if it's every year, but with some frequency. And last year's recipient was David Deemer, and in conjunction with John Barrows. David does origin of life research, studying the origin of life in shallow pools by creating self-replicating RNA-like structures, whereas John Barrows was doing deep sea thermal vent research. And so the two of them combined shared the award. And then recently, David Deemer was also honored being elected as a fellow of the American Academy of Inventors for his nanopore sequencing device. So he has several claims to fame. So congratulations, David. Are there any other announcements like that where we should give kudos to people for various accomplishments? Anybody online? Okay. Let's see. Another thing I wanted to tell you is that we have indeed finally signed our Space Act agreement with NASA Ames, which is very exciting. So that should help to facilitate programs that lead to new collaborations and the exchange of students and postdocs. So, and makes badging easier so that we can have more visitations. So we're going to begin a dialogue with NASA Ames to try and figure out how to leverage this Space Act agreement. So if you have ideas or things that you feel would make your research a lot more productive, let me know. For example, we all know that graduate students are very expensive. And in fact, we're starting to decrease the number of graduate students that we're admitting as a result. So additional funding for graduate students would be very welcome. Okay, why am I going to look to NASA Ames, a government institute that is even more squeezed probably than we are, for funding for graduate students? Well, I'm going to do that because NASA Ames has internship programs that fund graduate students for the summer period, for example, and even that would be a huge help to us. So I'm going to try and talk to the Ames management to maybe allow a few slots for Santa Cruz students for those kinds of programs. And the same applies for undergrads and graduate students. So anyway, let me know if you're interested in being involved. So that's one thing. Another announcement is that in October of last year, we submitted a proposal to the Dean's Office for a minor degree program in astrobiology. So that is working its way through the chain. And there are many negotiations that are underway to figure out who's going to do the advising, et cetera, and how that's going to work. But in conjunction with that, we put in proposals for 6 new courses. 2 of them are being taught this quarter. Ruth Marie Clay is teaching cultural astronomy, which is Astro 49, and I'm teaching Astro 116, which is our first upper division kind of fundamentals of astrobiology class. And I think there are some students here today from that class. So welcome, we're really happy to have you here. Yes, seems like there was something else, but it must not be important because I have forgotten it. So that's enough from me. I want to now bring up Miriam to actually introduce our speaker. And then, of course, after the talk, feel free to stick around and have a social hour and meet everybody, including our speaker. So again, welcome.
Hi, everyone. Welcome. Thank you so much for being here. My name is Miriam Tellis. I'm an associate professor in Earth and Planetary Sciences here at UCSC. And it's my pleasure to introduce Dr. George Cooper, our guest speaker for today. Dr. Cooper works at NASA Ames. Prior to NASA Ames, he got his PhD in chemistry from Arizona State University. Spent a couple years at SETI and then went to NASA. And at NASA, he works on these really complicated organic material in meteorites. And that's what he will speak with us about today. So his talk titled Interstellar Grain Molecules, Radiation, Magnetism in the Formation of Enantiomer Excesses in Meteorite Organic Compounds. Thank you.
Thanks, Mary. Okay, and if Miriam and everyone gets together, they can get me an office in this nice building here. That's why I put it up there. It's better than government. So the next slide is not moving. What makes it advance?
You should just go to the right.
Right. Yeah, you mean hit the
Right.
right? That's
The
what.
button is not working.
Yeah.
Neither is the return button.
What about this?
Oh, it's okay. You can just click. And then now
Yeah,
it's
there's
set up.
also these.
Oh, so those are better. Okay. Okay. It's not moving again. Oh, well, okay, now the other, okay. Yeah. Okay. So, the good news is I won't keep everyone here an hour. So, and some of the results I presented before and, showed to David, Miriam, but I'll show you what's new with them. So anyway, here at Santa Cruz, everyone has probably seen something like this. Organics are formed at some stage of the evolution of planetary systems, and they're formed on grains. Radiation magnetic fields are at play, although hardly anyone talks about magnetic fields, what that has to do with anything. But certainly we know that radiation creates organics. So my talk is going to be adding that in. And, let me just say it now, in case anyone's interested, I mentioned it to Dr. Tellis and Pran, who's online. Someone's probably heard of the recent controversy about racemic amino acids from the new asteroids, Bennu and Regu, and how this one unusual one is racemic, and therefore all this enantiomer excess stuff in meteorites the past 30 years is just garbage. So I have one last slide at the end that I tried to pack everything into, so if anyone is interested. Okay, so again, most people here know there are a variety of organics found in these 4.5 billion year old rocks, amino acids, carboxylic acids. I'll be talking about this particular class. And this is, sort of a somewhat familiar textbook stack of sugars. Added oxygen to that aldehyde group, and I'll be talking about that, the carboxylic acid. So instead of glyceraldehyde, you'll see results with glyceric, so instead of erythrose, erythronic. The meteorite parent body is oxidizing, so maybe these were oxidized. But I have this here because I'll keep referring back to the carbon number. So the next couple of slides, I'll say 3 carbon glyceraldehyde, 4 carbon, 6 carbon. Everyone's heard of glucose. In this case, again, the acid form. But this is just to show how rare the rare enantiomers in red are pretty rare. Something like D-aldose has never been found in nature, according to the last report. So 5 out of the 8 sugars, or again, the sugar acids, are pretty rare in nature, and that's going to be a point I'll come back to. Okay, so again, everyone probably knows the importance of chirality. in life, DNA and RNA only has one mirror image and not both. It couldn't, and the L, again, is pretty rare. And again, if you have cats, you can leave now because you already understand. What influenced the chiral properties of meteorite compounds? Did they lead to enantiomer excess? And let me just say, you want an enantiomer excess. This is the ultimate enantiomer excess. Only one enantiomer is in DNA, RNA, proteins only have one mirror image of amino acid in its structures. That's the ultimate enantiomer excess. So for a couple hundred years, people have tried to make at least one of these enantiomers a little more abundant than the other. Sounds simple. That has been a goal, since past year. Just do whatever you want to do, make one a little bit more abundant than the other. Okay. So also, how general are these processes creating enantiomer excesses? And down to us, can we duplicate an enantiomer excess in the lab? And so again, I'll start next couple of slides with glyceric acid, the 4 carbon twins, erythronic and threonic, and on down. Okay, so what led up to this? So this is not lab work. This is what we found in the meteorite years ago. The 3C, the smallest, is really 50-50 enantiomer, equal DNA mirror image. We went to the 4 carbon, and you can see here, it's the same for both. This one wasn't separated at the time, but 3 or 4 to 1, the D. So racemic D moved down to the 5 carbon, and it gets even weirder. A big D, small L, this is 7 or 8 times more. Okay, move down or up, whatever, to the 6 carbon. And this is just one of the unusual ones. I ignored gluconic and galactonic because that scheme I showed you, 3 glucose, galactose, and mannose are common on there. It could be a contamination in your test tube. So I don't even worry about it, although I actually believe those results. But here is one of the rare ones on Earth. It's the same thing. The D is predominant, and we couldn't find any L at all. So 50-50, 3 to 1, 5 to 1, and what? 17 to 1 we extrapolated? We don't know. So that's just one of the rare ones. Here are a couple more. Telluric is very rare on Earth. The L's in parentheses is not there. That's what we were saying. The same for another one, alike. Again, the sugar of this, the D form, is not found on Earth, but it is here in meteorite. Okay, so no student dropped their lunch in my test tubes, for any of this. So, okay, what are we going to try to duplicate what we see in the meteorites? And there's only been one sort of mechanism doing the same thing. They used inorganic complexes and got only enantiomer excesses of 10 to the -4 difference. So it's not talked about very prebiotically, but it's a good program. What we're doing though differently, we're putting a sample either here or there, irradiating magnetic field at the same time. I didn't want to put up all the details, but paraformaldehyde, a sample of paraformaldehyde, mildly alkaline, pretty simple stuff, and irradiate with, again, very mild radiation. Most lines, we have a mercury lamp. Most lines are indivisible. One is 365 nanometer, which is just barely in feet. So that's what we're doing. And so here's, I showed this before a while ago at LPSC, and poor Dr. Teller, she's seen this a million times, probably tired of it. But when we did that, we got an L excess and erythronic, and a D, less D. In threonic, you can see a large excess, enantiomer excess, but do you believe it? So reverse the magnetic field, so plus B, north to south, whatever you want to call it, the opposite turn the magnetic field around, and you had better reverse this enantiomer excess, and you can see here it does. D to L, and here D is dominant, here L is dominant. So that was like I said, we showed this before, and it was a good result, except I have not sent that to an editor. Why? Because even though we showed that, it is really, drives you crazy. You have to wait, do experiments, and I don't know, maybe 4 months, you will see it again. All the other experiments, they will be perfectly equal. In other words, no result. So the reversibility was consistent, but not EE. And again, 5 months later, aha, we did it again. D, L is excess, reverse your magnetic field, D is excess. So that was good, but why did we have to wait so long? Enantiomer excess. Did I?
Oh, so it just means, yeah, so you would do
Sorry.
the same experiment and you would get the same.
Well, right, this was the same as type of experiment, maybe minor difference as this one. But the point is, we have to wait months, so I couldn't send that paper in, because Miriam asked me about publication. I couldn't, can you imagine sending the result in and telling the editor in the accompanying letter, well, we could get this result twice a year, that's good enough, publish the paper, just leave us alone, publish. No, you have to be able to consistently do it. Now, what kept me going was, how could it be a fluke or contamination when you do get an excess, EE and that, you did get the opposite with the other, turn your magnet around, you did get the opposite. So it's like a gambler in Vegas, they just give you a little bit to keep you going, right? So I didn't quit, I didn't retire. You could, in another 6 months, do it again. All the others are failures, you would see that again. So what the heck was going on? So that is the new stuff I'm going to, the magic ingredient that I'll talk about that made this consistent. But first, I should just, again, I know people have seen this, critical to the story is just remember chirality. If you have a carbon, 4 different groups in mirror image, you could probably get differential effects, affect one enantiomer more than another, and that's the key here. So imagine these are locked in a magnetic field, and the photons, the catalysts are coming from the right. Well, you're going to get a reaction here first, over here, over this one, and before that one. That sounds fluky, but a science paper, I have it somewhere on my computer, just put H atoms here, the H2 molecules, all right, pretend that's an H and that's an H. The paper showed, and I forget the author's name, but I have it, they showed that when they're oriented towards the radiation, this hydrogen is ionized first. And the paper, the point was to show the difference in time from ionizing this hydrogen to that one. It was like 10 to the minus 18 seconds or minus 21 seconds. It's remarkable they could measure the difference. So I just say this, that had nothing to do with that, just to say that orientation makes a difference. And I'll get to that later. And one old example is by Bonner, who was at Stanford. Same thing, a D and an L mirror image in space. This was solid phase, so they're locked. And they use circularly polarized light, right or left, right, to see if there's decomposition. This is UV CPL, circular polarized light. Of course, both are going to absorb ultraviolet light and be destroyed to some extent. But here, and I won't go, you don't need to go through this whole table. The result was in one polarized direction, you had about 51% of the S and only 49 of the R. In other words, it destroyed a little bit more of the R. So that is an enantiomer excess. Again, that's the goal, and that's why, there's not many papers at all that do anything like this. So this is not magnetic fields, and other groups have done circular polarized light. And they reverse their their CPL. And so then to the left polarized light, and then the other is destroyed, So a little bit of an excess there. And so that was a nice experiment. Again, Origins Life people say, well, if you got to destroy 99% of all your organics, you get an excess, but we wouldn't be here today. So, but it was a nice concept. Okay, so on to what we're seeing now and why we could probably say we're consistent is I'm starting actually with paraformaldehyde, but formaldehyde. Whenever you have formaldehyde in water, You don't have that. You have the hydrated form. 99.9%. It doesn't matter if formaldehyde is here on Mars or in your bathtub with water. This is what you see. This is what's actually is not stable. But it's not chiral, right? We wanted a carbon with 4 different groups. So, that's old news. It's not chiral. But on interstellar grains, there's deuterated formaldehyde, there's deuterated water. Actually, in some cases, with D2O or 1 deuterium there, DHO, there could be more of this than normal light water. There's a lot of deuterium on grains, and I know some of you, probably have measured some of those grains. So when you do that now, instead of this non-chiral, theoretically, you could get a chiral formaldehyde, diol, whatever words you want to use. Now, like I said, it's not stable, but here, if this happened on interstellar grains, I would bet that it did. You're talking 5 degrees Kelvin, 10 degrees above absolute zero, with just about anything is stable, right? And remember, I didn't point out on the other slide, formaldehyde is what cascades, makes the larger sugars. 2 carbon, 3 carbon, glucose. So formaldehyde, the old Formose reaction, is a starting point for sugar synthesis now and probably 4.5 million years ago. But anyway, so again, deuterated water, you could get a chiral molecule. So what happens when I added deuterated water? So remember, years of kind of sort of consistent, not really. What happens? Okay. Oh, I should point out that once formaldehyde, appears, you can make, especially photons, glycoaldehyde to 2 carbon. And it can also have, it's not chiral, neither one has a carbon with 4 different groups. But again, add deuterated water and even glycoaldehyde, not chiral, can become chiral, theoretically. Theoretically, but I will bet you anything that I don't think astronomers, I don't know if maybe Natalie or someone could say if they've looked for any weird such things on grains. But even here, you can have a chiral. This is important because you don't need photons with glycoaldehyde to make higher sugars. You do it from aldehyde. Glycoaldehyde by itself will make all of these. You go in the lab, put in some glycoaldehyde, make it alkaline, go do whatever students do, rob Fort Knox in your spare, whatever you do, come back and you'll have sugars. So that's why the chirality is important. And maybe someday someone will search for these weird things on grains.
Sorry, really basic question. So can you remind us why there's so much deuterium, the interstellar medium?
Oh, the geochemists would answer it better, but the explanation I always read is you have an interstellar grain, you have hydrogens hopping around, you have deuterium, right? In the universe, it's not very abundant, but on a 5 degrees Kelvin grain, deuterium is just as likely to be stuck on organics or on the grain as a hydrogen, because the energy is not much different at those temperatures. That's why they're enriched on deuterated species like water. The lighter species are enriched on grain. And when you look at meteorite isotope compositions, the organics are all heavy in deuterium relative to Earth, always. And the traditional explanation is, on grains, a carbon will bond with the deuterium as easily as a hydrogen. Plus, with radiation and further reactions, DH bonds or DC bonds will fragment less. They're more stable than the hydrogen bonded. So that's another reason that for the enrichment.
You say that it becomes chiral when you use the deuterated water. It's not that molecule itself, it's the down products, the sugars that are created out of that formaldehyde that's made of deuterated water.
Well, this is probably real. It has to be when, like in our test tube, we use this instead of maybe a little bit of water. You have to have that, even.
In order to create those chiral.
Oh,
products.
well, these don't care what form this is in, as long as it's formaldehyde and alkaline solution or glycolaldehyde. So,
the chiral molecule has to have 4 different bonds to the carbon. That's why it.
I guess I just don't understand why it's chiral, like with the amino acid that you showed before, I get that D and.
if you make a stick model, just like the amino acid, make a stick model or the organic chemistry kit
Okay,
and have 4 different groups around carbon
oh, I see,
and make the mirror image,
right, they're not all in the plane, they're doing.
you cannot superimpose it at all.
Okay, thank you.
The atom, you can line up maybe this one and this one with your stick models, but the other two, the OD and OH, aren't matching.
Got it.
Right,
Thank you.
yeah, so again, you care because of force that differentiates one mirror image versus the left, like I showed, that might happen. Okay.
Got it.
So chirality is, and according to Jerry Joyce at, Scripps, David, I guess he's still there, right? Jerry, you can't have DNA and RNA, or DNA if you start with both enantiomers or ribose. They've tried it. They put an equal racemic mix in the test tube and they only get 4 or 5 units of DNA. You know how DNA repeats over and over. It just doesn't go. So you have to start with an excess, or he said that you have to start with an excess of whatever amino acids or sugars. He didn't care about space and molecules. He was just, doing his own research. So it's now, this next slide I'm going to show you, maybe this didn't do the trick, or maybe that didn't. But here's what finally, after 100 years of doing this, here's what happened when we add deuterated water. That same molecules before, 3 ionic D and Al, reverse the magnetic field, and you can see that the enantiomer excess switches. Now, I showed that before, right? I said it wasn't, but now you can get out, jump out of bed and do that, and you'll get the result. So now we get it over and over again, adding deuterated water. And for chromatography people that care, so here's another experiment, same, deuterated water. One magnetic direction, Al is more abundant. In this case, there's psi. This is the companion experiment. Switch magnetic field, and now D is more abundant. So chromatography people will appreciate this. We did a lot of these back to back, same DCMS, close in time. So you don't get contamination worries or something. So we did it, this same magnetic direction, south to north, did it again, and same result. Okay. Then went back, go to the conditions where you don't get enantiomer excess, you get 50-50 slice I was talking about before. Take out that deuterated water, and we had deuterated formaldehyde, so what? Now it's back like it should be to 50-50. So these, and I forget which order, but this might have been run at one time point, and this one an hour later, and that one an hour later, and that one, just for people, who does GCMS here? Oh, love the GCMS. Okay. Well, stuff you get trapped in your lines. You do an injection, you think you have a result, but I've seen stuff from a week earlier, still stuck in those lines, they come off and interfere, even though we clean it between. So anyway, that's what I said, that's inside chromatography stuff. Do them one after another. So now we're pretty convinced. And those were, I didn't want to write all the conditions down. We used calcium, in the alkaline solution. I think the complex is forming, I'll show you later. Without the reagent that causes EE, does magnetic direction alone show different chemistry? And sure enough, it does. So black is formaldehyde, red is C13 glycolaldehyde. In one magnetic direction, so plus B or north, most of the products come from formaldehyde. The black traces here, so 3 ionic. Okay, formaldehyde was regular 12 carb, C12. Switch to magnetic direction again, and red. So the glycolaldehyde now is producing, more of the products. So and they're not there's no excess here because we took out, in this case, calcium that is probably the agent doing part of the trick also, in addition to doing water. So, so erythral, so, again, that's the sugar, here's the sugar acid, but same result. The 4-carbon sugar in one magnetic direction, formaldehyde, black trace, and switch your magnetic field, and you can see the red goes up. So, and again, I'll show why, but I think there's some sort of maybe complex form where glycolaldehyde is in the right position relative to radiation rather than formaldehyde. And this is just one many experiments we did without touching the sample, no sample handling. Irradiate it, put it in an absorbance spectrometer, see what you get. And when you have two reactants, in this case two, they split. There's different absorbers, probably different abundances. We can't see inside cuvette. With one reactant, there's no difference between north and south magnetic directions. So with two, and so that's why, and again, I can't overemphasize, no untouched, breathe on the samples from photolysis, go to the spectrometer. So it's just to show that something, apart from the GCMS data, something is happening with magnetic direction. Okay, and so here's what the first pitch, you can imagine the calcium in there. So maybe, probably we're forming a complex when it's irradiated one direction. Like I said before, you get a certain product, switch your radiation or magnetic field around. It's the same thing, you know, switch, the complex is being irradiated first in the other direction. The calcium we have is almost certainly doing something. Years ago, Agnew was, again, not thinking about space and enantiomers, they just know that calcium forms very strong complexes with sugars. And that's probably in your textbooks now. Well, biochem, calcium binding, they're very stable. An LC column is packed with a calcium complex of sugars to separate sugars. That's how stable that is. So maybe, again, in our test tube, we're radiating from this direction, you get maybe the D excess and switch the magnetic field or the photon, same thing around, and maybe you get the other enantiomer. Maybe this is sort of what's happening. Okay, and so, you know, when did this happen? Well, I'm guessing that since these newly formed solar system, once this is more Natalie's bag, but once you start to get homogenized rotating fields with charges, with ions, you get the magnetic field. So perhaps early in this process, grains with the organics on them are exposed maybe to a neighboring star. We formed around a lot of giant OB stars, and I'll show you a Hubble photo next. But so add the magnetic field and radiation. Is this what we're reproducing in the lab? And, you know, could that be a mechanism for billions of years ago? Okay, so ALMA observation. So this is a drawing, obviously, but this is a real Hubble photo showing how giant stars today, as we speak, are irradiating newly forming solar system. We are here. This is us. At any one time, maybe the predominance of radiation is from one direction. Although, of course, you have other stars, you know, but if one single source is more important at any moment, maybe you can get excesses. The Hubble photo shows the tail of the forming solar system pointing away from the giant star up here. That's how some of these newly forming planetary systems are being blasted. Is that a scientific term? Being blasted to smithereens by giant stars. And I think what 100 or 200 stars contributed to our solar system, giant stars, at least from the meteorite data. So, well, I don't need to summarize this. So we just have more analysis working. If I ever get our 50-year-old GCMS units fixed, then I can send this paper out. I need 2 more runs. It's a conspiracy, I think. So what time is it?
406.
Okay, 406. Okay, so I told Miriam, we talked before, so it's a separate subject, but has anyone heard of the, I don't know, I hate to call it controversy, the Bennu results, the amino acids were receiving, including the non-biological amino acid isovaline. Has anyone read about that? Oh,
What is racemic mean? What is racemic?
oh, okay, 50-50 DNL. Right, so the results came back and papers published over the last couple of years show 50-50, like I showed a couple. And therefore, they said, well, this casts doubt on all the amino acid, not the sugar stuff, but all the amino acid enantiomer says is found over the last 40 years. So in case you do run across this, I'm talking about it at AppSciCon. They said that these amino acids, and they're more rare, but with a hydrogen attached to the chiral carbon, they're saying they're racemic in Bennu and Rigu. And again, we don't, the amino acid data from meteorites says that sometimes these do have an enantiomer excess. Well, not mentioned, and I'm not quite sure what's going on here. All of these amino acids with an H like alanine, they've been shown to be racemic also in regular carbonaceous meteorites. And that was used as a point to say, cast a little doubt on the last 30 years. And so they've always been shown racemic. So the one amino acid that's hard to racemize, in other words, you have a hydrogen here, it can come on and off. So if you start with an L amino acid, when that comes off, it forms a planar complex. The hydrogen can come back on the other side in equilibrium, and therefore you get the other enantiomer. So it racemizes, comes to 50-50. Geologists use that to date some rocks, how much of the other enantiomer. Here, the enantiomer is hard. You cannot normally racemize isovalent because that methyl is there. It doesn't pop on and off. So if you start off with an L excess, chances are you still have an L excess. So this was measured in meteorites for years now, as I said. The recent measurements in Bennu and Rigu show it is racemic. And again, that was used to cast doubt on the past 30 years, but it's also racemic in Murchison. Sometimes from grain to grain, it jumps. So what about going to something, to an analog, a larger analog of isovalent? This is 5 carbons. So Cronin and Pizzarello look at the 7 carbon in meteorites. So isovalent is rare in nature. Forget that. These have never been seen in biology form, but they're in meteorites. So this is actually, if you move the CH3 here, that's another compound, it's an isomer. So it's actually 2 compounds and another one in this paper. They also have L excess, like isovalent does sometimes in Murchison and other carbonaceous meteorites. This was not pointed out the last 2 years when the doubts about enantiomer excesses in meteorites. These, they're so rare, they had to make them, which is fine. You can make amino acids if you stick to it. But then when they got these peaks here, okay, cover up the D and L. If it's never been seen in biology, how are you going to know what's the D and what's the L? Right. So this is one compound here. Move the CH3 up here. That's the isomer. They had to go through long drawn out procedures to determine what was the L and what was the D. And I think they ended up using an enzyme from all kidney or some, you know what you biologists do. You go weird places and find things to find answers. And they did. Again, I'll talk about this at AppSciCon, but with this doubt business in 97, people should quote this. You can't go to something not found on Earth And I believe isovalene results in Murchison sometimes as an excess, most of the time, sometimes it's not. The 2 Bennu sample says it's not, but does the surface have anything to do with it? These people here, back in 79, irradiated isovalene in the solid phase like it would be on an asteroid, in the salt form like it would be on asteroids. And what they say was not much radiation cosmic, it racemized it. Is that what they're seeing in these 2 asteroids? Because they have to be on surface. When you sample Murchison, maybe the reason sometimes you see an L excess and sometimes you don't, maybe Murchison represent pieces from more the interior and the surface. And I maybe I'll throw it up at the meeting is people don't understand why, even to this day, why you measure and get one result with an organic. You go a few microns in the same sample, you get a completely different. You can have oxidized also and reduced. But maybe that's why they had to stick to the surface in these 2 asteroids. There's no choice. And from what I'm reading, again, maybe someone here probably knows a lot better than me. From what I'm saying, if you go down a foot, or 2 or 3 in an asteroid, the cosmic rays are still, Natalie, you are still pretty strong.
How deeply they penetrate, no.
Yeah. Okay, so, but, I would sort of tend towards that as the maybe reason, which means that if someone had, well, I guess this theory, get a deeper look at these 2 asteroids, maybe again, you would see an excess. But, and I didn't bring the trace of isovalene where Pizzarillo, she took, just 2, just isovalene, pretend that's isovalene. The L is in excess. At UC Davis, they did isotope measurements at a bunch of points on one peak for C13, trace each peak to make sure it's not, again, bunch peak, contaminating, which you won't see with isovalene anyway. But they did the same thing, another sample with hydrogen, trace each peak to see if that D to H ratio was high, like it is in space. And they also did the nitrogen, the NH2 group, so the hydrogens, the carbons, and more than one paper. So I can't just toss all that work, all those years aside and say, because these 2 show racemic that, well, there was no extraterrestrial enantiomer excess committed. So we'll see. If a mobster came to me and said, okay, are there an excess in isovalene in these 2 meteorites, despite what they're saying, I would have to say yes somewhere. Yeah. So we'll see. Okay, so well, either I died or talk is over. So I guess that's it.
I really enjoyed your talk, and I found myself very curious about this latter part of the talk, because when I think about a particle moving through space, I would imagine there's relative charge on the surface of the particle that could be leading to these racemic mixtures on Bennu and Ryugu. So I'm curious, like in your experiments or in the next steps you're taking, are you going to be looking at the actual like geochemical charge of the surface of some of these particles to see if that would lead to some of these responses in these rare amino acids?
Well, right now, we're just doing the mundane, boring stuff of analyzing both meteorite and the experiments. I need a bunch of grad students to branch off, and that would be interesting. But nailing this down, and again, the reason I'm not into too much else, this simple difference, if it's real, the DNA, again, 200 years, that's all people have wanted. They didn't have peaks back in 18, they just knew there was something asymmetric, but at least the last 100 or so years, this is all they want. So we want to make sure and nail that down, but that would be,
Yeah, I mean, I think it's really cool. This idea links back to your earlier experiments where you're flipping the magnetic field, right? Like the point of those experiments are if we change the chirality of these particles, are they going to flip back when we like have these like products from radiation, right? Like, are they showing the same signal? It's like, that's kind of the focus of these early experiments, right?
Yeah, well, the particles, I don't,
And I guess the particle part I'm mentioning is because I'm wondering, like, if you have these like amino acids on a particle, the local charges of the particle might really matter for the
Yeah.
irradiation products.
yeah,
It,
the charges make magnetic fields.
yeah,
You know.
Like even-
this, yeah, so I think that would, I'm not sure if the charge on the particle itself, I mean, that could be right. I mean, this could all flip in another planetary system. There may be L, sugars and people made of L DNA. So whatever charges there are, again, I don't know if the particle would, how much particle has to say about it.
that's
Yeah.
just
I
something.
guess just building on that,
Yeah.
like, we know that there's lots of mixing
Oh, yeah. Yeah.
to produce chondrites, or you're finding this. So we know that there's like material coming from very cold regions where you might have lots of excess deuterium, deuterated water versus material coming from regions with
Mhm.
less deuterated water. So you mix all of those together, then it could explain part of why you've seen so much variation on the very small scale with the meteorites.
But coming from a warmer region, I wonder if there would be enough formaldehyde left over. Or,
I guess yeah, that's true.
yeah, I wonder if, how much would be left over to cascade into sugars if it's too hot. And the D to H measurements of everything else is high. I haven't measured, I measured the D to H of glyceric, of the C13, but not the DH. So they,
I got a comment about Bennu and then several questions about your lab work. I understand that Bennu is basically a rubble pile.
Mhm.
So we all agree to that. That's a nice solid piece of rock and I don't think it originated in a rubble pile. It was a collision between a group of asteroids and a pretty good chunk of stuff that got to the Earth. Not as Bennu stuff did, they have milligrams of material to work with. So amazing work, by the way. I know all those people that did it, and it's really extraordinary work. But if they work with something different, something that is just put together as a big mixture from probably several asteroids that were involved in a collision process. And that's really different. I would say that jumping to this conclusion that they did is too much. I think there's so many other explanations that you want to work through before doubting the enantiomeric excess in your system.
yeah, there was another group, I didn't put the name up in PNAS, who had another mechanism for producing excesses, but on Earth. So they really sort of said, well, it had to be, forget space, it has to be all Earth happening. So Danny Glavin, he is, let me back up. Danny Glavin has done a lot of measurements of isovaline and Jason at Goddard, a lot of good work analyzing the receiving core, non-receiving nature. Even he, even today, he says, well, maybe these past experiments were, a fluke. But again, I wish someone would, which I'll do at AppSci, would point to this monster and his partner. And there's a third one in the same paper, if you guys want to look at it, with these excesses. So yeah, it is kind of early to be saying, based on what couple of samples.
Okay, that's your procedure. This is a mercury arc lamp, is that right?
Yeah.
And would it be the 254 nanometer frequency or wavelength that would be the primary activating wavelength getting into your system.
Yeah, the wavelengths are 264, 404, 436, 547, and 589. So the what did I say? 365, barely into the UV.
Yeah,
Yeah, it doesn't go as low as 254, although I have done experiment with 254 nanometer separate lamp, but this mercury lamp has those fine. So,
you know, my question is, given 254.
oh, okay.
Where in the molecule do you think the actual absorbance of that photonic energy is occurring? Because in order to do what you're showing there, differentiation into L and D and D and L, with respect to a magnetic field, in order to do that, you've got to activate some electrons within the fluid
yeah,
by
yeah.
absorbing photons and going to an excited state, and then it can relax into the other, for instance, the other enantiomer.
Sure. David, I might have the measurement we did on the same USB where, for example, glycoaldehyde. Oh, well, right here. For example, glycoaldehyde or formaldehyde with those lamp energy, 365, 404, they do nothing. They don't absorb. Add calcium. And again, I don't want to say too much for folks, but add calcium. Now you see a difference. I have the absorbance, what you're talking about. I have the absorbance spectrum. The absorbance spectrum of glycoaldehyde has been produced, shown for years. Not too many when you add a calcium. Then you get absorbance in the range of our lamp. So, you know, if I had to say, it would be a calcium complex of this and maybe formaldehyde too, but you know, I know for a fact that this absorbs, so it is becoming more energetic.
So whatever happens to the electron structure is being altered when the calcium is binding. And that calcium is not binding to whole charges of a sort of the equivalent of hydrogen bond.
Complexing, yeah.
Yeah, complex.
Yeah, yeah.
Last question is, what was the strength of your magnetic field?
You mean something else that I didn't want to say today? Most experiments, it's been around 3,000 or 3,100 Gauss. I have results now that waiting to be analyzed at 1,600 and 400 Gauss. But even the 3,000, even though that sounds strong relative to space, the meteorite matrix only shows a Gauss at the last measured magnetic field in meteorite shows 1 or 2 Gauss. So our 3,100 is a lot. But I looked up the equation for polarizability, and I don't have that. If you extrapolate from room temperature down to 5 or 10 Kelvin, then you don't need 3,100 Gauss. 100 or 50 Gauss, maybe even lower, would suffice in space because the temperature is orienting. Remember, we want orientation here. We want these to be oriented. So at 50 Kelvin, they have to be more oriented. So probably if I extrapolate it right, 25, 50 Gauss could have done it, and then transient higher magnetic fields from shocks or whatever at any one time. So yeah, that's the first thing I worry about, and a lot of other papers suffer from, was how prebiotic is this? So
Is there an estimate of the magnetic field strength in the stellar system?
yeah, that's the 1 or 2 Gauss.
1 or 2 Gauss?
That-
Okay.
Well, it's 1 or 2 Gauss is the last measured magnetic field of the minerals in most of these meteorite, right? Miriam, oh yeah, so that's the last measured. I've seen papers that hint that they could have been in it when any one time 100 Gauss happens, and then when the minerals equilibrated in the lower, so I would have to ask her.
So you're using a stronger field just because that lets you do the experiment.
Yeah, and let you write papers and get grant money. But I'll see what happens with the 400 Gauss, if I can get my instruments working. And then when you extrapolate down to 10 Kelvin, I mean, you would have to say it's certainly possible, I would think.
So if I understand this slide correctly, and correct me if I'm wrong, you've got small molecules, complex and is small, right? And it's polar, it's spinning, just as things spin, so it has a dipole moment, and that's why it's oriented to the field, and then you're facing one particular side of it to the radiation, and something happens that's different depending on which side it's facing. So am I interested in that? Correct?
Yeah, well, it's, right now, it's just a guess.
Right, okay, yeah,
But,
so
yeah.
that's correct, then that's small things floating by themselves, right? So should I be thinking about this happening before they become a solid, or?
I think in the, oh, I can just touch this. No, I can't. Okay, I gotta go back. Oops,
Like while they're floating in space, and
yeah,
you make the solids out of things that are already kind of.
I think here, before, it has to be transparent to radiation if this model is correct. So you can't have too much clumping yet.
There are a lot of dust grains in there though. Like,
Yeah.
what we're looking at there is little dust grains, they're already in molecules big, so is it happening before you make the grains, or?
Well,
Like.
I would say any place that the organic on a grain, even though if it's, I don't care if it's a millimeter or a centimeter below the ice level, that's fine, anywhere it's exposed. So at any stage, I would think that you're getting too late to create enantiomeric excesses when you get into these larger bodies.
But you need it on the grain, right?
Where can expose, yeah.
Calcium is solid. It is great grain.
So then
Okay.
it's not like a free floating thing with a induced dipole that's why is it reoriented to the magnetic field if it's like in a solid? This is a basic question.
Well, even calcium carbonate, which it actually is formed in our reaction, even that has been shown to be susceptible to a magnetic field. The grain sizes formed are smaller or larger. They've used that for hard water treatment for decades. They've used magnets on your pipes at home because the calcium carbonate that's forming the clogs is susceptible to magnetic fields. So here, calcium, they were, complexed. I would think as long as it's, I would think, so again, put calcium right here. I would think as long as it's sort of oriented in a magnetic field, it doesn't matter.
Yes, I'm just showing my ignorance, like, I know that like grains, if they have the charge, they will spin that, and then they will orient to the field, but aren't they still like a lot bigger than the picture you've got here? Like, so orienting the whole grain, it's not obvious to me why that would orient the small little molecules.
I'm wondering, like I said, I don't know what stage, but I'm wondering, when you first get an organic that's sort of attached to anything, maybe it doesn't have to be a big grain. If the radiation is not too harsh, and you would think it's susceptible. So the grain, I couldn't.
Progress.
Yeah, I couldn't, I couldn't speculate. Yeah.
Thanks, this is super interesting, I learned so much.
Yeah, sorry, I'm gonna need geochemists and physicists to talk about grains. Yeah, this is just pure lab chemistry extrapolated.
Yeah, well, I do have a question about, the abundance of some of the amino acids you showed us in the biosphere. You said one of them was really infrequent and the other one was basically non-existent. So yeah, why is that? It's just a matter of temperature. You put this amino acids in temperature, they turn to sugars very quickly. And maybe related to that question is how stable they are, say, to processes like planet formation, or if you were to crash one of these asteroids with like into like Europa, would they survive, or you would expect them to just get destroyed in the process of crashing against the Earth?
Yeah, the fact that they're in meteorites returned to Earth.
Yeah.
Centuries, literally. They are there, and weird ones are there. They survive.
Right.
Like Murchison, I think, and David probably knows better than me, but I think it air burst from the pressure. Some was shot back and floated down gently. But we have this in samples in all museums, and so survival is not the issue. Um, the stability in general, in the mildly alkaline conditions of the meteorites, these are pretty stable. If you do too much with base, you destroy them.
Okay. And so if you imagine that the Earth was bombarded by meteorites at some point, why do we don't have like a lot of them?
Or at what, at some point?
If you imagine like the Earth was bombarded with meteorites, right, at some point, like.
Even tonight, today,
Right, you know what I mean, like
stuff is
2% of the mass of the Earth, right?
Yeah.
Something like, so why don't we have a lot of them today?
a lot of the meteoritic.
All these guys, yes, isobiolite,
Well, biology doesn't really create these, it just doesn't need it. The meteorite is non-biased, abiotic. If you give it the amine, the starting amine and ammonia and small carbon molecules, if the energy is there, it's gonna make it. But biology does not care, about some weird guy like that. It just doesn't need it if I'm.
oh, yeah, no, yeah, but I guess my question was. If they are meteorites.
Mhm.
And these meteorites come from, say, asteroids, the asteroid belt or whatever, and that material also impacted the Earth when it was forming, right?
Mhm.
And that's significant matter to 1 or 2% of the mass of the Earth, right?
Mhm.
Why wouldn't they survive to the present day, right? Because we don't see them, right?
I think the geologists say the Earth was hot when bombardment was
Okay.
molten at one point. After Cruz, this had to survive, but life probably ate the original amino acids that came from space a
Okay.
long time ago. So the meteorites are preserved. So yeah, between a hot Earth and early life, I doubt you could find anything, from way back then.
Right, so they can survive then an impact, but not a magma ocean stage.
Oh, right. I think that's the general
Right.
thinking, yeah. And then the later bacteria. Was experiment 1910, someone fed a dog instead of the L amino acid, they fed dog the D. And, he was just fine. He didn't use it. And, but, yeah, if this stuff was around, it's all been consumed by bacteria. They don't care. As long as there's a carbon there they could use. So no, it's no way. On another planet, we have to have dogs with L protein.
Wait, so if you
Yeah,
find them on Europa, then maybe there's life?
Oh, covering up the life, who was, no, Pran. No, you too, you more were, nitrogen fixation, but Pran was, I don't know if Pran's online, but
I'm here.
yeah, did you hear his question?
I don't know. But first,
Oh.
let's,
That's,
We do,
Oh.
we do have a question online.
Oh, is there a
that's been
question online?
waiting.
Okay,
Yeah,
so-
Artem, do you want to ask your question?
Yeah, thank you. Thank you, George, for the great talk. I have a somewhat basic question, is perhaps a clarification. So, this enantiomer axis is meant to explain the right-handedness of life on Earth, and if so, my understanding is that you said this is also due to the irradiation from nearby stars. So that will suggest that all life in the solar system, including that we could potentially find in icy worlds, would have the same right-handedness, but then would that be the same on other on exoplanets, basically? Could you clarify?
Right, that, oops. No, I'm alive, yeah, there. If. If this is model is correct, where you have radiation coming in one direction, and all of these disks have magnetic fields, that's just known. Why in another planetary system can you have predominantly radiation from the other direction? So if we're correct, life in the universe has to be 50-50, you have 50% D ribose and 50% AL just from magnetic direction. The trouble with a nice, model that you that you like and extrapolate that to the planet. The problem is it's probably dead wrong when we talk about 4.5 billion years ago. But that's the working hypothesis now. The important thing now is we have to get this work in, or we're gonna get fired or something. But I'll think more about that, you know, as time goes on. But yeah, it should be random. That's a short answer.
All right, let's thank George for a wonderful time. And of course, we have maybe a little bit of time before he has to run off. If you have questions, you could ask him before he leaves, and there's snacks in the back.
After the refreshments.
Thank you everyone for being here.
Mary,
Thank you, everybody online.
could I have a pointer? I could use it for LPSC. No. Oh, thought I did.
Afterward, but that was great.
Oh, thanks.
Based on progress. They learn from, I got family stuff at home, so I didn't say, oh.
Yeah, thank you.
So you were so.
Okay, yeah, thanks for coming today. Yeah, I hope we have this out